Biology Part 101, Propagation of Nerve Impulse. The term propagation refers to the process whereby the nerve impulse travels along the axon of a neuron without decreasing in size in a non-myelinated axon, such as the squid joint axon. The impulse travels smoothly along. This process is illustrated in the figure. The part of the axon involved in the impulse at any one time can be considered in three sections. There is first an active zone where the nerve impulse is at peak. At this point, the axon positively charged inside. As a result, small positive electric current carried by ions in the cytoplasm of the axon flow into the adjacent negatively charged region of the axon. And across the axon membrane, ahead of the peak impulse, this positive current flow acts in the same way as a stimulus pulse, and the depolarizes the next part of the axon called depolarized zone to such an extent that it reaches and passes its threshold. When this happens, the depolarized zone becomes active and produces its own impulse, so that as the process continues, the impulse passes along the axon. Behind the impulse in the refractory zone, the axon is temporarily unable to become active, and consequently any current flow from the active zone has no effect. This is why the impulse normally travels in only one direction, namely from the cell body down to the axon. If resting axon is artificially stimulated somewhere along its length using microelectrode, the nerve impulse travels off in both directions at once because there is no refractory zone to prevent it. The impulse in a typical non-myelinated axon normally travels at a rate of about 1 meter and or less than that, the speed of the propagation, also called the conduction velocity. It can be increased increasing the diameter of the axon, where speed is particularly important, as in many escape responses joint axon have developed. Thus the joint axon, the squid, are involved when it uses its siphon for a jet propelled escape. The nervous system of many other invertebrates also contain the joint axon. In vertebrates, the conduction velocity is greatly increased by the presence of insulating material around the axon of many neurons. This is the function of the myelin sheath as shown in the figure. Electric current can only leave the axon at a node of Ranvier where the axon is uncovered. The region between the nodes does not produce an impulse but allows ordinary electrical conduction as in a wire. Thus the distance between the nodes is crossed almost instantaneously, and the impulse jumps from node to node it travels along. This process is referred to the statutory is referred to a saltatory conduction and allows conduction velocities of up to 100 meter. Myelination is effective even for small axon, and as a result, invertebrates' nervous systems can contain many more cells than invertebrate systems. The combined advantage of the efficacy and efficiency of the economy achieved by the myelination have allowed the vertebrate's nervous system to become extremely sophisticated, culminating the intricate machinery of the human brain. Well, the ionic basis for resting potential and nerve impulse. The resting potential and nerve impulse both depend on the distribution of particular ion across the neuron cell membrane. The most important of these ions are the potassium ions K+, and the sodium ions Na+. Well, the table lists all the internal and external concentration of these ions for the squid joint axon. And as a result of the radical differences of concentration, there is a strong tendency of potassium ions to diffuse out of exon and for sodium ions to diffuse in.